My own work on developing engineered arteries, which then became human acellular vessels, or HAVs, really began when I was a postdoc in the mid-90s, working for Robert Langer at MIT in Boston. I became very interested in the potential of this new field called tissue engineering to recapitulate structural tissues that could be clinically relevant for patients. And part of this motivation really came from a particular case that I did while I was training as an anesthesiologist at Mass General Hospital. What occurred in this particular case was a patient who needed a heart bypass operation, but who had no suitable vein of his own. And this led to the surgeons doing multiple incisions in his body to try to harvest artery or vein from different body parts in order to perform his heart bypass operation. And watching this case unfold really drove home to me the importance of being able to use the biology that we now know, both developmental biology and adult reparative biology, and using those biological principles to coax cells to make tissues that can be helpful for patients in the future. So I really began the work sort of with that premise in the mid-1990s. We published about 4 years later, in 1999, our ability to grow animal-based vessels, both bovine and porcine vessels, and to implant them autologously into animals. But this autologous regenerative medicine approach is something that we pursued for a number of years. But as we started considering what it would take to go to the clinic, it rapidly became clear that harvesting cells from a patient, going back to a factory and making an artery for that patient over a process of 3 or 4 months, and then returning to that patient, that model was going to be very difficult to scale if we were going to take it to many thousands of patients in the future. And so we became very interested in whether or not we could use cell banks to engineer human arteries and then render them non-immunogenic. And so Humacyte spent several years developing a decellularization process whereby we could grow arteries from scratch using vascular smooth muscle cells that are human and that are seeded onto a biodegradable scaffold. And after growing those arteries for a period of 2 months, we worked out methods using a combination of salt solutions and detergents to wash the cells out of the tissue. What that allowed us to do is to produce a tissue that now, after 8 or 9 years of clinical experience, has been implanted into over 450 patients without any evidence of clinical rejection. The additional advantage of having a non-cellular product is that because it's non-living, it has a shelf life. So currently our shelf life is 18 months for our human acellular vessels. So really finding a way to use a cell bank and then to generate a product that is non-immunogenic and that has a shelf life has proven to be some really important cornerstones of our ability to move our clinical trials forward and also, frankly, to build the value in the company.